Hello everyone, welcome to BioBusters. In this video, we will cover everything you need to know for subtopic 2.8, Cellular Respiration, for topic 2, which is Molecular Biology of the IB Biology HL curriculum. I'll begin by introducing what is cellular respiration, and we will also talk about ATP, the types of cellular respiration, those being aerobic and anaerobic, and their steps: glycolysis, the link reaction, the Krebs cycle, and the electron transport chain. This will be a brief explanation. In the next video about topic 8.2, it will be a better explanation. And then we will also briefly talk about rest parameters. We can first start off by saying that all living organisms obtain energy from food. The nutrients in food, such as carbohydrates, are broken down into glucose, which is absorbed into the blood and taken up by the cells. That is where the actual energy generating process takes place, which is called cellular respiration. Cell respiration is the controlled release of energy from organic compounds to produce ATP, which is the fuel our bodies use in the form of adenosine triphosphate. This ATP is made in the mitochondria of the cell. The main organic compound used for this process is called carbohydrates, as we mentioned before, which is glucose. Glucose is a preferred energy source from the brain cells and is broken down in the muscle cells for energy. Glucose is also the most widely used source of energy in all living organisms. Cellular respiration involves enzymes that control the process to ensure that energy is produced when it is required. And it takes place in two different compartments of the cell. Firstly, the cytoplasm and the mitochondrion. The conversions that take place in the cytoplasm are by the process called glycolysis, and the synthesis of the actual usable form of energy, ATP, takes place in the mitochondria. Now, ATP is a high energy molecule that functions as an immediate source of power for the cell processes. It is a molecule that carries energy within the cell, and ATP fuels most of our body's energy needs. And from cellular respiration, ATP is immediately available as a source of energy for the cell. Energy is produced when ATP is hydrolyzed. One molecule of ATP contains 3 covalently linked phosphate groups, which stores potential energy in their bonds. When ATP is hydrolyzed to form ADP plus Pi, the energy stored in the phosphate bonds is released to be used by the cell. Cellular respiration uses energy stored in these organic molecules to regenerate ATP from ADP plus Pi via oxidation. As mentioned before, there are 2 types of cellular respiration: anaerobic respiration and aerobic respiration. Anaerobic happens without oxygen, while aerobic, as can be seen in the name, includes the use of oxygen and its last step, which will be talked about later on. The process of glycolysis breaks down a glucose molecule, which is a 6-carbon molecule, into 2 molecules of pyruvate, a 3-carbon molecule. It is a metabolic pathway that gives a small yield of 2 ATP and 2 reduced NADH pluses and an H plus ion. Although the amount of ATP generated through the glycolysis is small, it is still better than having no energy at all during anaerobic phases. Both anaerobic and aerobic respiration pathways begin with the anaerobic breakdown of glucose in the cytoplasm, which is this glycolysis process. The glycolysis process includes 4 steps: phosphorylation, lysis, oxidation, and ATP formation. The phosphorylation starts with a hexose sugar, which is typically the glucose, the 6-carbon molecule, which is phosphorylated by 2 molecules of ATP to form a hexose biphosphate molecule. This phosphorylation makes the molecule less stable and more reactive, and also prevents the diffusion of it into the outside of the cell. The second step is the lysis, which the hexose biphosphate molecule, the 6-carbon sugar, is split into 2 triophosphate molecule, a 3-carbon sugar. The third step would be oxidation, which hydrogen atoms are removed from each of the 3 carbon sugars via the process of oxidation to reduce NAD plus into NADH plus H plus ions, which 2 molecules of NADH are produced in total, 1 from each 3-carbon sugar, which was also mentioned above. Finally, we have the ATP formation. in which some of the energy released from the sugar intermediates is used to directly synthesize ATP. This direct synthesis of ATP is called substrate level phosphorylation, and in total, 4 molecules of ATP are generated during glycolysis by a substrate level phosphorylation, 2 ATPs per each 3-carbon sugar. Now, anaerobic respiration proceeds in the absence of oxygen and does not result in the production of any further ATP molecules, only those produced in the glycolysis process. It occurs only in the cell cytoplasm as it involves only glycolysis, as mentioned before, and the process of anaerobic respiration is to restore stocks of NAD plus molecules, as this molecule is needed for the process of glycolysis. By restoring the stocks of the NAD plus via anaerobic pathways, the organism can continue to produce ATP via glycolysis itself. There are 2 types of anaerobic respiration, alcoholic and lactic fermentation. Alcoholic happens in plants and yeast. The pyruvate is converted into ethanol and carbon dioxide. And for the lactic fermentation happens in animals, in which the pyruvate is converted into lactic acid or lactate. The conversion of the pyruvate into lactic acids or ethanol and carbon dioxide is reversible. Hence, pyruvate levels can be restored once oxygen is present and a greater yield of ATP may be produced aerobically. And here on the screen, we can see the equations that the anaerobic process is produced. Now we will be talking about respirometers. A respirometer is a device that determines an organism's respiration rate by measuring the rate of exchange of oxygen and carbon dioxide. It is a simple apparatus that can measure the rate of respiration. Aerobic cellular respiration uses oxygen and produces carbon dioxide and water, and we can measure the consumption of oxygen as an indication of the respiration rate. The few steps of a respirometer are the following. As you can see on the screen, in tube A, the organism to be tested or germinated seed is positioned, and the tap is closed. The organism starts respiring, consuming oxygen and producing carbon dioxide and water. Then the alkaline solution at the bottom of tube A will absorb the carbon dioxide produced, whilst tube B is the control where no oxygen is used or carbon dioxide is produced because no living organism is present in it. The capillary connection, as you can see, connects the 2 tubes is a manometer, and the reduction in oxygen in tube A will reduce the pressure in tube A and will move the colored liquid in the manometer in the direction of tube A, providing an indirect measurement of the oxygen consumed, allowing the amount of oxygen consumed per time unit to be calculated. Finally, a respirometer can give us a good idea of the respiration rate of germinating seeds, a resting animal or a moving animal, and it allows us to compare different species. It is also a device that determines... And that is all for Subject 2.0 of the IB Biology curriculum. Thank you for watching.